Good morning. Today we will start our first lecture in medical parasitology by an introduction. Parasitology is the area of biology or science that's concerned with the phenomena of dependence. This means that one living organism depends on another for life, for food, for shelter, for anything. And there are many types of parasites. Some affect animal while others affect human. Here in medical parasitology we deal only with the parasites that infect man or human. We study the diseases they produce, how these men get the infection, and the response generated by human against these parasites. And for sure we will study or we will learn various methods for diagnosis and general outlines for treatment, and also different methods or regimen for prevention and control of this infection to prevent its occurrence from the start. Parasites are classified according to their location into two types. The first one is ectoparasites. Ecto means outside. This type of parasites live outside the host, only attached to the outer surface of the host or superficially embedded in the body surface or in the skin, like all or most of arthropods. That other type is called endoparasites. Endo means inside. This type of parasites live inside the body, either in the liver, in the lung, in the gastrointestinal tract, wherever. These endoparasites include all protozoa and helminthes and can be divided into three subtypes. The first one is called obligatory. Obligatory parasites mean that they are physiologically dependent upon their host. They cannot live without them, or they cannot survive if they are isolated from their host. The second subtype is called facultative. Facultative means that they are capable of becoming parasitic only if situated in a suitable condition. Otherwise, they are free-living organisms. The last subtype is called incidental. Incidental means that this parasite enters or attaches to the body of a host different from their normal one. Parasites are also classified according to their relation to the host into two types. The first one is called permanent parasite. These parasites live their entire life inside or on their host, but they cannot leave them. The other type is called temporary or intermittent parasite. They only take food from the host and they leave. Each parasite has its own and unique life cycle, including its host, its final habitat in the human body, its mode of transmission, its specific diagnostic and infective stage. Let us learn together the meaning of these terms. First, we will start with types of hosts. The first one is called definitive host. It is the one in which sexual reproduction of the parasite takes place. While the second one is called reservoir host. It includes any animal that harbors the adult parasite. So, definitive and reservoir host harbors the adult stages or the sexual stages of the parasite, but definitive host is usually a human, while reservoir host is always an animal. Also, from epidemiological point of view, reservoir hosts are very important in the control of parasitic diseases. The third type of hosts is called intermediate host. The intermediate host is one that alternates with the definitive host, in which asexual stages of the parasite are found. What's meant by habitat? It is the site in human body in which the parasite is located. By other means, its home, its preferred place inside the human body. There's another term called vector. It is an agent, usually an arthropod, that can transmit an infection or a parasite from one host to another. This transmission may be mechanical or biological. Mechanical means the parasite is transmitted without undergoing any multiplication or development inside this vector, while biological means that the parasite is transmitted and undergo multiplication or development inside this vector. How can the man get the infection is called mode of transmission, and each parasite has its own infective stage that enters the human body and causes the infection. So what about the diagnostic stage? It is the stage that when I see by microscopic examination, I can confirm my diagnosis. When I see the diagnostic stage, whether in stool smear or in urine smear or in blood or in bone marrow aspiration or in lymph node biopsy, wherever, when I see this diagnostic stage, I can confirm my diagnosis. Types of parasites include helminthes, protozoa, and arthropods. Helminthes includes platyhelminthes or flatworm, including Trematoda and Cystoda, and nematohelminthes, which means cylindrical or roundworm, including nematodes. Let us take some examples of each of life cycle of a trematode and a cystode. This is an example of life cycle of a cystode called Taenia saginata, in which the human is affected or get the infection by ingestion of undercooked meat of this cattle. So from this life cycle we can conclude that man harboring the adult stage of the parasite is an definitive host, and the cattle which harbors the asexual stages of the parasite, which is the larva inside its muscle, is considered the intermediate host. This is another example of life cycle of a trematode. This child defecates on ground. He has the adult worm is in his intestine, so it is a definitive host. The larva is present in this fish, so this fish is considered an intermediate host. There are many forms of relationships between the parasite and its host, the most important of which include commensalism and parasitism. Commensalism means that it's type of relationship that doesn't involve physiologic interaction or dependency between the host or the commensal. In this relation, the commensal or the parasite, which is a commensal, feeds on substances taken by this host. So in this relation, only the commensal is benefited while the host is unaffected, and the two partners can survive independently. Each one can live alone without the other. While parasitism is a relationship between two organisms. Usually one is smaller than the other one, but the smaller one usually dependent or physiologically dependent on the bigger one. How this parasite can cause harm to its host? many techniques. The first one, mechanical injury. It can make pouring a hole into the host or digging into its skin. It can stimulate a damaging inflammatory or immune response that has a severe effect on the host, or it can interfere with the host's nutrition. There is another very important term called zoonosis, which is used to describe an originally animal infection that can be transmitted to human either directly or indirectly through a vector. So it is an originally animal infection but can be transmitted to human. In this year we have many examples for zoonotic diseases, including leishmaniasis, trypanosomiasis, and trichinosis. In the life cycle of any parasite, we have to know its mode of infection or its mode of transmission or sources of infection. How could this man get the infection? There are many modes of infection or modes of transmission, including contamination or penetration of skin. Let's know each one separately. Some parasites can be transmitted by contamination of food and water by the infective stage of the parasite. Other parasites can be transmitted by penetration of skin or mucous membrane, like farmers that walk barefooted in their farms, or humans on sea beach. Also, there are some parasites that can be transmitted by vectors or an arthropod, like malaria. Also, some parasites have direct transmission either by kissing or by sexual intercourse. Some parasites can be transmitted by inhalation. These parasites that can be transmitted by inhalation have sticky eggs that can stuck firmly to dust particles that will be inhaled by human. Iatrogenic infection by organ transplantation or by blood transfusion. Also, congenital infection, that means vertical transmission from the mother to her baby. Finally, autoinfection, in which the human can reinfect himself. In each parasite, we have to know some main items. Epidemiology, which is the study of distribution and determinants of the disease, means its geographical distribution, its presence all over the world, determinants and risk factors for presence of this infection, if it is more common in males than in females, or the reverse, or it's more common in some races. Then morphology, shape of the parasite and its characteristic criteria under microscopy. Life cycle, which includes the habitat, the diagnostic and infective stage, the different hosts, the mode of transmission, as we discussed before. Then we have to know the pathogenesis of this parasite, the capacity of this organism to produce the disease. How can he produce the disease? And this depends on many factors. First, intensity of this infection, the number of the parasites that enters the human body, size and shape of this parasite, the mechanism by which the parasite causes damage, whether it causes mechanical damage or inflammatory damage, and the immunological response of the body to the parasite. After pathogenesis, we have to know the clinical picture of this disease. How could this patient be presented? Symptoms means what the patient feels. Abdominal colic, diarrhea, fever, sweating, and signs that are what the doctor discovers by examining the patient, whether splenomegaly, hepatomegaly, jaundice. When a parasitic infection is suspected, I have to confirm my diagnosis clinically, laboratory, or by imaging. Clinically, by taking history from the patient, whether he has lived in an endemic area or visited an endemic area within the preceded period, and by clinical signs and symptoms as discussed before. The second item in diagnosis is laboratory diagnosis, which includes direct methods, indirect methods, and detection of the parasite DNA or RNA. Direct method means detection of the diagnostic stage by microscopic examination, like detection of different stages of malaria in blood film, as we will discuss later in this year. Indirect methods mean detection of the parasite antigen or antibody using serological test or immunodiagnosis, like using ELISA technique. Detection of the parasite DNA or RNA using molecular techniques as PCR, which is polymerase chain reaction. The third item in diagnosis is imaging, including X-ray or CT scan. After diagnosis, we have to treat our patients either by drugs or surgical intervention. And finally, we have to prevent occurrence of the disease from the start and use different methods for controlling the spread of this disease. By treatment of infected patients, control of vector if present, proper hygienic habits in case of parasites that are transmitted by oral route, chemoprophylaxis if present for some parasites. Thank you, my dear students.